Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm excited to be joined today by two really excellent guests, Dr. David Dismuke and Dr. Stephen Gray, to discuss what it takes to translate gene therapies from the bench to actual scalable therapies that reach thousands and hopefully someday millions of patients. We're talking about a topic that's really important and sits beneath the surface of every gene therapy conversation, which is manufacturing in the cell and gene therapy space. So David's a recognized leader in AAV bioprocessing and GMP production. Stephen's professor of pediatrics at UT Southwestern Medical Center and also the co-director of the gene therapy program. David's the chief technical officer at Forge Biologics, which is one of the premier CDMOs that is behind some of the most impactful gene therapy programs today. So I'm excited to have this conversation today. So your paths have crossed, I think, a number of times over several decades in some unexpected ways. David, I wonder if you could start by telling the story of how you and Stephen initially worked together and then bring us to the present where you work together at Forge Biologics, you as CTO and Stephen as scientific advisory board member.
Sure. So again, very, interesting paths that we've crossed, but we both started off at Vanderbilt University, getting our PhDs there. It started the same year, in a collective program, kind of went our separate ways within that path for our PhD. But then we both ended up in Jude Samulski's lab without any sort of communication, just independently, and got through my interview, and I was like, I think I know you. Steve started there before me, and I'm like, and he's like, yeah, I was at Vanderbilt. So we had that sort of same connection, having some same classes at Vandy. And then ending up in Jude's lab together and working on some co-projects while we're graduate students, and then postdoc students, and then I went on to run the UNC Vector Core, and Stephen went on to be a professor, assistant professor, and we worked on some additional programs there. So I'll hand it over to Stephen. So
yeah. It was kind of crazy when I forget if I was like a year or two in my postdoc at Jude's lab at UNC, and then then David comes to interview, and it's like, you look familiar. Like, how do I know? And making the connection, but even at that point, I mean, I think that David did some research in the lab for a little while, and then it wasn't too long before he started moving to the AAV production side and doing some amazing work there, and then I kept on the lab side and was working together, and the first GMP product that I ever needed to have made, David really spearheaded that at the UNC facility.
So Stephen, maybe tell me about your academic research today. What do you work on at UT Southwestern? Give people a sense of the kind of therapy areas and research program, and then David, I'd love to hear the same for Forge Biologics for people who aren't familiar.
Yeah, so this is, kind of the culmination of about 20 years history of working with adeno-associated virus or AAV vectors, engineering them, optimizing approaches to try to target the nervous system, and work that I started at UNC, and then moved to Texas, UT Southwestern, about 8 years ago, to it's essentially, you can think about it as like a platform kind of approach, platform technology, where you've got an AAV vector. We've optimized approaches to get it to deliver it to the nervous system, and then just applying it to one disease after another disease after another disease, and then building a sort of bench to bedside pipeline where we're doing preclinical drug development and gene therapy, and then moving things over into clinical trials, navigating through manufacturing, toxicology, regulatory, all the above.
David, same question to you. A little bit of a background of Forge Biologics and what you all do as a company.
Sure. So we're a contract manufacturing organization based in Columbus, Ohio. And we really started about 5.5 years ago with an idea to develop more of a platform process for AAV manufacturing. Been in a number of companies, and it was always start from scratch every single time. And it was difficult to for every client when I was a client to have to go make a new cell line, to create a whole new process, and actually took sort of a learning from our UNC days. So when Steve it was a client at UNC, we had basically a platform process. People just came and plugged in to what we did, and it was relatively simple, simpler for the client. And so we took that same approach, but scaling it out much bigger, and, a lot more GMP rigor, and to try and make it simpler as a starting point for AAV manufacturing, and also to kind of move away from using research tools to make AAV. I think our early days, when we started, we were using plasmids and vector manufacturing systems that were really set up for that were research tools that we just applied to AAV manufacturing, and so we took a step back and tried to see how we could redesign them for improved manufacturing, improved product quality. So my oversight at Forge is really on our R&D group, our process development and analytical development teams, as well as just oversight for the company. So we're about 450 people, and we have about 60 clients that we produce AAV for.
And so, David, in my mind and in many of our listeners, AAV manufacturing, we think of these big bioreactors, vats, you're growing viruses. Yes. Can you just talk through the process? What is it like at a high level from beginning to end to go from you've got a design for a vector, a candidate design, and you're bringing it through all the way to actually putting it into patients to help? What does that process actually look like beyond just the visceral image of these enormous bioreactors?
Sure. So it all starts with the cell, right? The cell is basically the factory within the factory that produces AAV. And there's really 4 main manufacturing systems. You can use baculovirus, SF9 insect cells. You can use producer cell systems, or you can use the herpes virus system with BHK21s or HEK cells. We really use the transient transfection method. So transfecting HEK293 cells and big reactors. Yes, that's certainly true. Mostly in the AAV side, everyone uses single-use reactors. So you have a plastic container with inside the stainless steel shell that you're growing your cells and then adding the DNA. I think when we started out, production, for AAV, the yields were fairly low for HEK293, but I think over time, ourselves as well as others have improved those processes so you grow the cells up till they get to a certain point. You add the DNA, basically the instructions for making the virus, the vector, and the gene that you want to insert. And then usually, 3 or 4 days later, you harvest that. It's a terminal process. And then then this goes through the stages of purification, usually affinity chromatography, followed by ultracentrifugation or ion exchange chromatography, polishing steps to get to the final vector in the vial. So,
and Stephen, maybe a related question to this. You talked about AAV for a number of different brain disorders. Can you help me and the listeners understand how much variability there is vector to vector between any given set of programs, and what does that feedback loop look like, both in the manufacturing step, but also learning the longer outer learning loop of, was it safe? Did it work in the clinical trial? And how do we then feed that back into vector design? I just want to get a sense of, is it possible to get to a universal brain vector? Is there some subset, and where are we at today?
Yeah, well, and David will probably chime in, or might chime in to say every product is always different, and it is true. But the two basic components here are, the viral capsid itself, and that can be very uniform, and its properties to kind of target where the virus goes in the body, and the doses that you're using, and most of the manufacturing components are going to be suited to that capsid, right? And that's going to be pretty static, and that's the platform on the delivery side, platform on the manufacturing side. But then where everything is customized is the cargo that the capsid carries. And, if it's a gene replacement strategy, then you have the gene, but there's also the regulatory elements, the promoter, the poly-A, different, 5 prime, 3 prime UTR, however you're designing it to regulate, because when it's delivered, it ultimately will end up in the cell as an extra chromosomal episome, and it's going to function almost like a mini chromosome. So it has to be an independent transcriptional unit. So that's where all the customization happens, and that's where some of the manufacturing properties can vary from one vector design to another, and that's whenever we're applying them to diseases. Some genes that we deliver, we can express at very high levels, and there's never a problem, and that's it's fine, and it's simple, and it can be pretty generic. Other genes, we deliver them, and if you express them in the wrong place or at the wrong levels, they can be toxic. And so you have to carefully design those vectors to make sure that the capsid is taking it where you want it to go, and then the packaged DNA is expressing it the right way.
And a question for both of you, really. Maybe I'll hear from you, David, first on this. What we learn from the manufacturing process itself, you're probably looking at how many of the capsids are empty, what the efficiency and purity of the process is, but also we want to know, is this going to have the desired effect in patients when we get there? And what can we learn from trials that vectors that may have been designed 2, 3, 4 years ago that are being deployed now? How do you think about the feedback loop that's within this whole process?
Yeah, so I mean, I have worked a lot, in therapeutic companies, and as well. So I've actually done a lot of what Stephen's talking about as far as design of the transgenes and trying to come up with improvements. And I do see a lot of movement there, right? As far as companies, if I can list out names, but like Form Bio, who's trying to do this, through an AI approach and a more high throughput approach. I think it's more difficult for AAV than it is for some other types of therapeutics to employ those tools, because at the end of the day, you tend to need to manufacture at some scale to get that readout. But we're always trying to have better manufacturability by the design. But of course, for the therapeutic approach, you need to Stephen's point, need to make sure that the promoter works where you want it to work. And we obviously see a lot of movement in that area. But I also see a lot of movement from our clients coming in on novel capsids. So I think, and that creates a new challenge for manufacturing, because now it's slightly different in how you need to purify that vector. And to Stephen's point as well, every vector is a bit different. So the manufacturing is certainly different, but then we can't forget about analytics. And so that creates a new set of challenges around what analytical assays you need to develop specifically for that product. Typically, it's ddPCR for understanding the dose, and then analytical ultracentrifugation or other complementary methods for understanding empty full. And then importantly, what the potency assay is. And that can take quite a bit of time depending on the approach that someone wants to take, whether it's a relatively simple readout of RNA, or it's looking at the protein of interest that may be generated, or actually looking at a functional potency assay that's typically required for a licensure.
Yeah. And if I can jump in really quick, you kind of talked about this feedback loop. And this is something where, when we're using, say for a class of diseases, we're predominantly using one capsid, then as products are developed and they go into the clinic, and you're starting to get some of the clinical readouts from multiple trials using the same vector, then you start getting feedback in terms of different adverse events, serious adverse events, and also efficacy signals, coming both ways, and maybe some of the trials where you might expect that it might work, and then for whatever reason, it's not, then you might be looking back at some of the design of the drug and the dose and the potency. But also on the safety signals on the other side is, if when a serious adverse event happens, then one of the things that people will go back and look at is the manufacturing. And, what are the attributes of that drug? Was there any of those attributes that might have contributed to it? And that's where, when David's talking about empty and full capsid particles, we dose based on full particles. So the potency of the drug, for the most part, is going to be the same whether you have a lot of empty particles or not. But the safety can be affected, because if you have a lot of empty particles and you're already dosing pretty high, then the antigen load that the body will see, can be a lot higher with a product that like has a lot of empty capsids versus something that's relatively pure. So that's kind of a learning over time, that I think, where you have this interplay between, clinical outcomes and manufacturing.
Yeah, and David, is that ratio, for example, full versus empty capsids, is that a function of both the vector and the manufacturing process itself, or is it mainly driven by the manufacturing process? How do you think about that?
No, it's definitely an interplay, right? So we see a lot of very complicated transgenes. So AAV has a packaging limit of about 4.7 kb, and I guarantee you that's a pretty firm limit. We do have some clients that we can produce over that, but the efficiency is very low in producing, say, a 5 kb transgene and packaging it. So but then you also have different types of microRNAs that have secondary structure that makes it difficult for packaging, or repeat promoter elements that get truncated during the process. So design certainly impacts the manufacturing, but then the manufacturing also impacts what that product quality looks like. And so typically you're going to make, depending on your process, between 10 and 50% full particles through your production. So now you have to enrich that subpopulation through your downstream processing. And that can be used, there's a variety of different ways. You can do gradient ultracentrifugation, which can be very good at separation of those, especially for like cesium chloride ultracentrifugation, because you can actually see the particles, but it's a labor-intensive process. Where ion exchange chromatography, a more high throughput method, a more industrial friendly method, can also achieve that. But then you have to make a sacrifice, usually around yield. It's less more difficult to remove the intermediates, which is, we kind of talk about full, we kind of talk about empty, but then there's partial packaging, there's high molecular weight packaging. There's a lot of different particles that get produced, and so you really want that full particle population, and that's going to be impacted by how you want to purify it, what's your strategy, but also the design of the vector overall, as well as even the capsid will come into play.
There's a big downstream consequence of all of this, which is cost as well. Was a recent 60 Minutes piece that went into a sad case of a young child that needed a gene therapy. I think the price tag was $2 million. Insurance wouldn't pay for it. We know that with genetic medicines, the stats suggest that there are much higher success rate in clinical trials, but the trade-off we have today right now is they're an order of magnitude or more, several orders of magnitude more expensive on a per dose basis. How do you see that? How have you seen that cost curve come down in the time you've been working? And then how do you see that going forward? Because there's a lot of different numbers depending on the organ system and how much you need and the complexity. It can be $1 million a dose, but it can also be 10, 20,000, right? Getting into the range of a biologic therapy or other kind of modality. So it'd be great to have both of your perspectives on that. Who wants to take it first?
All right. Well, not to the extent that David has, but I mean, I've run an academic manufacturing AAV manufacturing core, and have some idea about raw material costs and everything, and they're substantial. So there is a real cost to manufacturing these drugs, and some of it's raw material costs, some of it is expertise, and I can let David go into that a little bit, but keep in mind this is right now there's I don't know the number. There's maybe 10 FDA approved AAV products, give or take. So we're kind of in a first mover situation where the products that are moving in right now, those companies took on an enormous amount of risk, and they're using cutting edge technology that's still really being worked out. So the costs, understandably, are incredibly high. The costs associated with manufacturing those drugs and developing those drugs and factoring in the risk that those companies had to take. So, I think that with any kind of technology, the further it gets along, then costs are going to come down. And I think one of the big drivers of that is kind of innovation on the production side, where, okay, the raw material costs may be similar, but you can make 10 times more out of a batch. And I think that that's where over time we're going to see costs come down, and also where the AAV capsid technology is getting literally orders of magnitude better. There's some incredible innovations that have happened, and so if a manufacturing batch produces 10 times more, and then a capsid is 10 times more efficient, you can lower the dose. Then all of a sudden you're talking about like your manufacturing costs dropping by a couple orders of magnitude. So in the long run, I think I'm very optimistic that things are going to get a lot better. And I think you've seen that with other technologies like monoclonal antibodies, for example, where when they started off, they were incredibly hard to manufacture, incredibly expensive, and now they're super cheap to make. So
they're strikingly similar headlines from the 80s and 90s, right, that we see today.
Yeah. So I mean, we're in a period right now where I think it's incredibly painful, and but I'm optimistic that it's going to get a lot better, and I feel the pain because a lot when you're talking about gene therapy, it's almost impossible not to talk about rare diseases.
Yeah.
And so you have a lot of these patient populations where there's just not a huge market in terms of patients, and so it's hard for drug developers to kind of move into that space, just because there's not a big commercial market, and then throw on high manufacturing costs on top of it. I see the push and pull, because if the drug costs aren't high, then there's no incentive for the companies to get involved, and they can't recoup their costs. But on the other hand, I'm also dealing with the patients that have to deal with those high costs.
Yeah. So yeah, Steve stole all my thunder. One thing I think we can't is hard to take away, right? There's always obviously manufacturing that goes into this, but drug development in itself is a costly business. Developing the vector, testing and preclinical studies, which Stephen knows very well. And then obviously, clinical trials themselves are expensive. I think we see movement regulatory wise to lower those burdens, which I'm glad to see.
Yeah.
But there is still, of course, manufacturing piece. And again, Stephen's stealing my thunder, but we've gotten much better at one capsid design so that you can lower the dose. I do see that shifting away from as less natural serotypes, which can have lower efficiency, to more engineered capsids, which have higher efficiency. So you can maybe potentially drop those doses 10, 100-fold or more, depending on your route of administration. And then, manufacturing wise, what we've been trying to do, we're not the only ones, but really trying to go back to the drawing board to see how can we, redesign these plasmids, redesign them, engineer the cells. And what we've seen, we tend to talk about AAV again around the dose, about how many full vectors you have and how much you're producing on a per liter basis. By scientific notation. So typically when we started, we were in the high E12, E13 VG per liter. and now we're really approaching, mid E15 VG per liter. So you're talking about 100-fold improvement in, over our careers and what you can produce from AAV. And a lot of this has taken place in the last, 5 years. And so being able to produce it at high titer, having better purification strategies, so you get more of what you produced. So we have clients who came in and said, I need to produce, a 500-liter batch or a 1,000-liter batch. We were able to reduce their batch size down to a 50-liter. So that's, the cost of the materials that go in, for the cell, or for the cell media, for the other reagents, that doesn't change, but the scales come down. The other thing that we're seeing is, a lot of treatments that are being now developed for non-rare disease. And so it puts things in a whole other, pickle. How are you going to produce, for diseases that have millions of patients? And so that's, I think that's the new challenge. But what I'm very encouraged by is those, improvements that we're making for the manufacturing side to be able to produce it for millions of patients brings down the cost per dose, which actually helps a lot for the rare disease community. So I think that's a great thing to see the field kind of a resurgence going after some of these non-rare. And I look forward to seeing how that really drives more, patients that can be treated for rare disease.
Yeah, that you went right to a question that I had as we start to move into more common diseases like macular AMD, macular degeneration, Parkinson's disease. There are growing scaled clinical trials in some of these diseases that you could see gene therapies going to hundreds of thousands or millions of patients in the next decade. I'm wondering about this cost structure. Is it scaling to this level a marginal cost or a fixed cost point? Like, we have to build a whole new factory to do dosing on that scale? Or is it actually that a lot of the fixed costs you can just figure out how to improve the viral genomes per liter, and you get possibly pretty dramatic marginal cost improvements with the existing infrastructure we have? How should we think about how much the common disease demand might help this problem versus it, needing some viral vector design improvements that are, to the point you made earlier, Stephen, push that, another order of magnitude, say, on the number of viral genomes per liter that you can get to really see the cost efficiency that we need for this. What thoughts do you both have on that? David, I'll start with you since it's a follow-up to your question, then go to you, Stephen.
Sure. So I think there's a number of approaches there, as far as, how to, if you're going after, these millions of patients, it does change how you set up a factory, right? So we're typically built as a flexible factories with single-use components, because you need to change over to make another product, and you don't want to have any residual product from, and so that's a great strategy for these more rare diseases. But you get into more common diseases, I do think it shifts a bit in the scale at which you can produce, and it probably moves more back to more traditional manufacturing that you see for like monoclonal antibodies, where it is stainless steel, it's clean in place. And so it's, and maybe potentially, higher volumes where we get into the 5,000, 10,000, 20,000 liters. So that is certainly a doable approach. And if you're at that scale, you probably also take monoclonal antibodies are not made in a transient process. Typically, they're made through a producer cell line. And so those are strategies that would be, because then you can avoid the cost of plasmid. You can also, it's just easier to scale. So I do think that's happening, but, I'll kind of hand it back over to Stephen, because I think the route of administration, capsid design, vector design is also, it's been changing within the field and improving, and a lot of learning. Stephen's been a big driver of that. So, I think that's something that's going to continue to improve for these non-rare disease.
Yes, Stephen, and while you build on that, I would love if you could go back to the point you're making about AAV vector designs delivering maybe an order of magnitude better, because I'd love to hear a little bit about how the design of these systems is, what's going into that, how it's changing, what you're seeing. Is it natural serotypes that are being discovered, or is it engineered? And talk a little bit about that, because I thought that was really interesting. I want to make sure we came back to it.
Yeah, and I'll start off really quick by saying I think the manufacturing challenge is almost because it's somewhat divergent. You know, you have on one end the challenge of massively scaling up around one product, and that is a very different challenge than making, you know, many hundreds of little micro batches, if we want to call it that, for individual diseases and being able to turn those over and utilize a platform. They're two very different kind of engineering problems, I guess. But because right now, I mean, there's literally thousands of diseases that could potentially benefit from gene therapy, and you know how to turn those over and make those small scale batches for each one. But yeah, in terms of some of the newer technology, I'd say, you know, for most of the history of gene therapy right now, we've been relying on just naturally occurring viruses, and we're sort of capturing those AAV capsids pulled straight out of nature and hijacking them and using them as gene delivery tools. And we've kind of like been screening those and sorting through those and getting the ones that are the best. And we've gotten to the point that we are now, but now there's a whole wave of new engineered capsids with small modifications, small targeting peptides, for example, that, you know, I'm in the central nervous system space. So one of the big advances are capsids that will target receptors in the blood-brain barrier to enable very efficient crossing of the blood-brain barrier. You know, again, like 100 times or better, you know, than you know what we've been using up until now that has been used in some approved drugs. And so that enables us to be able to treat diseases better, but then also we can use lower doses, which end up being safer therapies and cheaper to manufacturing and everything. It's all good. You know, some of the challenges associated with that is every time there's a new flavor of an AAV, then you know, one, there are some unknowns, and there was a safety event that happened with one of these next generation capsids where a you know, first subject in a clinical trial passed away. And we all don't really know exactly why and how. But the other thing is we talk about platform approach, and you know, if David and Forge are used to manufacturing this one capsid and they've done a couple hundred batches with it, and then we come to them with a modification and something that's a little bit new, then they don't know if all of that transfers over exactly, or if they've got to kind of reinvent the wheel a little bit. You know, so it's always when you're in a platform, there's a lot of comfort, there's a lot of benefit of that. And then, you know, eventually you've got to move out of that to a new platform, and there's some growing pains associated.
As we look forward, are there areas of this that you all are particularly excited about? It's a challenging time for gene therapy, but the science is really good. We've also got gene editing. What is most interesting? CAR-Ts are moving beyond into non-oncology indications, immuno applications, autoimmune diseases, for example. There's a lot of interesting areas. What are you guys most interested and excited about right now? David, I'll start with you.
Yeah, I mean, obviously, Stephen and I come from a background of what we call traditional gene therapy, which is kind of an oxymoron in a way, but you know, where it's really about replacing a gene that is defective. And I think what is, you know, I think very intriguing for me is we're moving into all these different modalities, you know, probably with, you know, gene editing and, you know, prime editing being, you know, I think the cutting edge, and where things, in my, you know, opinion, you know, probably go, you know, a lot of diseases can be treated in that same approach. And to Stephen's point, it simplifies the process. It simplifies the product if it's basically the same thing each time, and it's an easier way to apply those platform learnings. So I'm very excited about what the future holds. Again, even though it's, you know, been a few decades of work, I think we're still just scratching the surface of what AAV can do, and what you know, so these, you know, applied technology. So, but I think eventually, yeah, you know, base editing, prime editing, you know, CRISPR base editing or other editors probably are the long-term future, and they fit pretty well, within an AAV delivery platform. So I'll hand it over to Steve.
So I always say gene therapy, the most important thing about gene therapy is delivery. And that was the case 20 years ago, and it's the case today. And all the advances that we've made, everything that we've done to get to the point that we are, I would categorize our ability to deliver the genes is in most cases barely good enough. And so we end up having to push doses high to try to meet the delivery requirements that we need to, and then we run into safety events, and then the manufacturing is expensive because we're delivering really, really high doses. So it's all about delivery. That's the most critical thing. And now with some of these new tools, some of these new AAV vectors, it's almost that I don't want to jinx us, but it almost feels like the delivery could be a solved component of this. And then that opens up the door to a lot of creativity of being able to do things safer, but also start to incorporate things like gene editing, base editing, where I think for the most part, the gene delivery wasn't efficient enough to even go there.
And now it might be. Yeah, is that because the vector design, you're just getting so good at targeting them to the right tissue organ system? Is that the unlock?
Yeah, I mean it's really like you have a certain particle dose, and then that'll get you a certain percentage of the cells, and then the dose is capped. If you get above a certain dose, then you start hitting toxic thresholds. So, and when you're doing gene editing, this is getting a little bit technical, but a lot of these end up having to be split vectors. The genome and the guide RNA and everything. Right. It's too big to fit in one AAV. You've got to split it over two AAVs. And then there's no way, like it's become so inefficient to get both of those in the same cell expressed in the right way. And then the editing itself can be relatively inefficient. So
you're stacking lots of probabilities together that you don't want.
Yeah. So it just becomes, with the current delivery tools that we had, a really uphill battle. But when we're talking about next generation AAVs that are 10 times, 100 times more efficient, then it just opens up the door to a lot of things. And I'd say that's what I'm more excited about than anything else.
Yeah, it's really interesting. You both spend time in Jude Samulski's lab, as we covered at the beginning. He is a legend of the field. I was really just curious to hear what you learned in the group. What were the big takeaways you had from that group? Because it must have been a really formative experience. I'll start with you, David.
Yeah, I mean, obviously, so many great scientists have come out of Jude's lab. Zhao Zhao and Doug, Arvin Asokan. So we've been blessed to have peers, that are around us that we're doing really good science, and following in their footsteps, quite honestly. So as far as Jude's mentoring of us, I think it was pretty profound, and his ability, especially when I was in the UNC Vector Corps, really understanding how to develop a drug, early days of AskBio, as they were working hemophilia. So getting sort of insight into that aspect of it, and seeing, quite honestly, around the corporate side, he's a phenomenal scientist who has this amazing recall of everything that's been done before us, but also how that is going to be applied. I think that's uncommon. A lot of scientists are good at seeing maybe the what's going on right in front of them, but how is it going to be applied and make a drug that works. At the end of the day, that's what you have to do. It's great to do amazing bench science, but if it's not a drug at the end of the day that works in a patient, then it's fruitless.
Yeah, Steven, I think I'll follow on that with the same theme of, one of the things that I learned a lot is looking at the big picture, like looking at the whole landscape and where the gaps are. And, sometimes people just pound and pound and pound away at something, but it's not the biggest problem. There's something else that's going to hold them back. And, that was around the time that David joined at UNC. I think Jude recognized, like thinking about treatments for Duchenne muscular dystrophy and the enormous amount of drug that would have to be manufactured, and that the manufacturing platforms available were orders of magnitude away from being able to scratch the surface on that. So, doing a lot of investments in manufacturing at UNC and developing those platform approaches really years ahead of what other people were doing. That's led to CDMOs and manufacturing platforms that a lot of people are using now. So I think that's something that I try to always carry with me is, making sure that you're focusing on the right questions to have the biggest impact.
Amazing. Well, I think you guys have done just that for us today. You've given us a lot of good questions to focus on, and how we can make this the decade of gene therapy. So thank you both for your time.
Thank you, Patrick.
Yeah,
Nice
this
to be
has been fun. Thanks a lot.
Thanks, everyone, as always, for listening, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.